everyone. Today we learn about botany, second year botany. Already we discussed the first question, that is RDNA technology, process of RDNA technology. The second question is, let us learn which tools may involve in process of RDNA technology. That we learn here. Out of these two, one question is compulsory. So everyone, you have to learn, you have to concentrate on this area. Let us see, already we seen the process of or steps involved in RDNA technology. Today we learn which things are involved, which steps, which tools are required for RDNA technology, that we see. In this, very simply, five major things are there, or five major tools are there. Number one, RDNA technology means we cut the DNA and we insert the DNA into the another wanted, another plasmid, then it have to be insert into the another host cell. For that we require, here, look here, for we have to cut the DNA. How we cut, we cannot see with our naked eye the DNA, so we are using some enzymes. For that enzyme, the number one, the first foremost important enzyme is restriction endonuclease. Number one, restriction endonuclease. Number two, polymerase. Number three, ligase. Number four, vector. Number five, host organism or host cell. Let us see first one, restriction endonuclease. Already we know what is endonuclease. Restriction means it may cut the DNA. The wanted DNA is cut by a enzyme. That enzymes we call it as restriction endonuclease. The DNA which is cut by a enzyme, that enzymes we call it as a restriction endonuclease. Okay, depending upon how it cut, depending upon where it cut, the restriction nucleases are two types. Number one is exonucleases, the second one is endonucleases. Ase means enzyme, restriction means cutting, where it cut, depending upon which side it cut. The enzymes, the restriction enzymes are two types. Number one, exo and endo. Exonuclease and endonuclease. Exonuclease means, for example, look here, this is the DNA. Okay, when the DNA is cut at the end of the, at the tip of the DNA, that we call it as exonuclease. The first one, exonuclease. Exo means the outside, endo means inside. Exonuclease means where the DNA it cut at end of the DNA, that we call it as a exonuclease. Exo means it may cut a tip of the DNA or ends, if it is either 5' end or 3' end. It may cut, it may have capability to cut the DNA at end of the DNA. That is exonuclease. The second one is, or B is endonuclease. Endonuclease means, already we people know, DNA means it consists of different types of the sequences. Okay, different types of the sequences, like look here, A, G, A, T, A, C, like that. Nucleotides are there. In that, this we call it as a nitrogen bases. Depending upon the nitrogen bases, different types of the nitrogen bases are there, but these endonucleases, it may cut DNA at a specific site, that we call it as a endonucleases. Look here, for example, this is a DNA sequence. Here the DNA sequence, it is cut at the place of the A and between G and A. Look here, at 5 prime end, it cuts the G between the G and A, that we call it as a endonucleases. Example for this endonucleases, EcoRI. This EcoRI is collected or extracted from the Escherichia coli. Okay, so it is the restriction enzyme, so that we call it as a RI. That name is EcoRI. Endonuclease. Example for this endonuclease, EcoRI is example for endonuclease. This is regarding first one, that is restriction nucleases. The second one, we cut the DNA, or we cut a wanted DNA. Look here, up to here we have a DNA segment. With only this DNA segment, one segment, we cannot do anything. We required number of the copies. We required Xerox copies. For that, we are using poly chain reaction, that is a PCR. In that PCR mission, we are using the polymerase enzyme. Poly means number of. Poly means number of. Polymerase means it is useful for the copying of DNA segments, or duplicating the DNA segments, or duplicating the wanted DNA. For that we call it, for that we are using polymerase is an enzyme. At the time of the how the DNA will be reproduced, how the DNA will be prepared, by process of replication. Here the first point is the DNA is the polymerase is the enzyme which involved in the replication. Next one, replication means the DNA copies are prepared. DNA copies are synthesized in PCR. Okay, in PCR, polymerase chain reaction. Next one, it is useful for the amplifying the fragment, or amplifying the wanted DNA. It is useful for the amplifying means we have to be synthesize new DNA segments which we have, that we have to we make a copies, we make a Xerox copies. For that, this polymerase enzyme may useful. The next one, the third one is the ligase enzyme. Ligase, in the sense, you will remember, it is a pedicle like enzyme, means it attach to wanted DNA and as well as plus vector. Look here, we cut the DNA here, we have a DNA segment. This DNA segment we have to insert into the vector. At the time, this wanted DNA, it have to be attached with the polymer, it have to be attached with the host vector. The polymerase is a enzyme which involved to attach the wanted DNA or desired DNA and vector. Very simple. It look like it is a act as a gum. It act as a attacher. It act as a binder. Okay, look here. It joins the two ends of the plasmid and wanted DNA. That is ligase. Ligase means a gum. which can able to attach the two DNA molecules or two DNA segments, that is wanted DNA, and which we have to be insert that wanted DNA into the vector. That is the main role of the linkage. The next one is the vector. Vector also a tool. Vector means, the first point, vector means which is lower molecular weight, it not belongs to the chromosomal DNA. Extra chromosomal DNA. Extra, we call it as a vector. The vector, it may help to the wanted DNA. It transferred into the host cell. It is a vector. Okay, that vector have a simple, it have different types of the characters. The number one is, it is a low molecular weight. It must have low molecular weight. Why? If it is a high molecular weight, the transfer should be very hard. It is difficult to transfer into the host cell. So that the vector, it should have very low molecular weight. Next one, the vector, it can be cleavage site. The vector, it should have cleavage site. Why? We have to cleave, we have to cut the plasmid or vector, then we have insert that wanted DNA into the plasmid. So that the vector, it must have cleavage site. Next one, the vector, it must have replication site also. Okay, replication site also. The vector, it must have selective markers. Okay, selective markers in the sense, already we seen in recombination DNA technology. In recombination DNA technology, we seen the selection of transformed cells by the media, selective media. At the time, these selective markers are very useful. With these selective markers only, we can select the transformed cells. Okay, that is also a characteristic feature of the vector. Vector, it should have low molecular weight. It have to be cleavage site. It consists what we call it as selective markers. Okay, it have to be ORA, replication site also it have. Okay, these are the vector. Next one is the last one, host organism. We require host organ. Now we have rDNA. rDNA means plasmid and wanted DNA. rDNA means plasmid and as well as, not only plasmid, any vector, and as well as next one is the wanted DNA. This vector and wanted DNA, we call it as rDNA. That rDNA, it should not be perform any work without host cell. So we have to insert this rDNA into a cell. That cell we call it as a host cell or host organism. In that host organism only, the cell or desired DNA, it can be active and it can be produce what we require. These are the tools of RDNA technology. These are tools of the RDNA technology. It is very important. Okay, very simple, and 5 things are there already, which is already you learn in RDNA technology, process of RDNA technology. So that it is a very easy. You remember this 5 item, 5 sided. Number 1 is the restriction engine. 2. Polymerase engine. 3. Ligase engine. 4. Vectors. 5. Host organisms. This is tools of RDNA technology, tools of biotechnology, whatever it is. The tools of RDNA technology is this. This is a very easy, and listen it and Thank you for watching. Please subscribe our channel. Please support us. Thank you, one and all.